Now we'll proceed to session 6, vascular disease across organs. To lead this insightful session, we are very honored to have Professor Helmut Augustin, Professor of Vascular Biology at Heidelberg University, and Professor Ronald Wang, Division Head and Deputy Director of Prenatal Genetic Diagnosis Center at the Chinese University of Hong Kong to moderate the session. So time to them. Thank you so much.
Thanks a lot. So, okay, so we keep the best for last, and we look forward to sharing this session. And we start with the first presentation. Where's our speaker? Okay.
So may I introduce the next speaker, Professor Jack Wai-Ho Tan. So Dr. Tan received a PhD from Lei Ga-Seng, Faculty of Medicine, University of Hong Kong, and he awarded American Heart Association postdoc fellowship to continue the study in Yale University Cardiovascular Research Center in the US. Currently, Dr. Tan is the head of the Department of Diagnostic Science and professor of School of Nursing and Health Science in Hong Kong Metropolitan University. So he is also a principal investigator of Guangzhou Medical University, and his lab is focused on platelet heterogeneity in cardiovascular diseases. So may I invite Dr. Tan.
So first of all, thank you so much for the invitation from the organizing committee, and thank you for the introductions. So I'm Jack Tan. Actually, my work is quite different from these 2 days talk about the vascular muscle cell as well as the endothelial cells. So we switch to the smallest cell in our body, which is the platelets. Okay. So platelets actually is a nucleated cell in our body. They are very small. They only have 2 to 3 micrometer in diameter, but they are very important. Why they are so important? So because for the platelets, they play a very important role in both physiological and pathophysiological situations. So in the physiological way, the platelet can help to do the vascular integrity. So they do the hemostasis, as we all know that they can form blood clotting, and also they can secrete a lot of growth factors to help the wound healing as well as the angiogenesis. But not only the vascular integrity, they can also help to improve the immune response by doing the inflammation, because there's actually a lot of inflammatory cytokines inside the platelets. For example, there's interleukin-1 betas, TGF-beta, TNF-alpha. They all can be found in the platelets. So that's why during the inflammations, there's a lot of inflammatory cytokines actually secreted by the platelets instead of the other inflammatory cells like the macrophage and also the lymphocytes. And also, very recent advance in the field already found that platelet can actually do the cell-cell communication through the microRNA transfer. So actually, platelets can work as a vehicle. They can transfer their microRNA contents from themselves to the other recipient cell, for example, like in the smooth muscle cell and also in the endothelial cells. So they help to do the cell-cell communications. So in the pathological situation or in the disease conditions, we all know that actually platelet is very important for the thrombosis formations. So that's why in many of these vascular diseases, for example, like the atherosclerosis, venous thrombosis, as well as some, for example, like DIC, so platelet can play a very important role. So up to now, the major drug that help us to prevent the cardiovascular disease or stroke is the aspirin. So that's why platelet is very important. And the other thing is in the disease condition, for example, like the infections, platelet can also play a very important role because, as I said, there's a lot of like inflammatory cytokine as well as the chemokine in the platelet. So that's why they help to recruit the immune cell and also do the inflammations. And very recently, there's a lot of paper published about the role of platelets in other disease, like in the inflammatory disease, kidney disease, as well as the liver disease. So that's why platelet is very important in our body. So, why platelets are so important and why they have so many different functions in our body? One of the reasons why is because the platelets have the heterogeneity. So that means they have a very subpopulation like many different cells. So recent advance in the single cell sequencing let us know that actually in our body, there are many different subtypes of cell. For example, like the vascular muscle cell, that's actually, we have already have a lot of knowledge about there's a very different subpopulation for the smooth muscle cell. But for platelet, actually, they also have a lot of different subtype. So during the lifespan of the platelet is very short, only like 7 to 10 days for the platelets. So they have from young to age, they will we call it the circulatory aging. So during this process, there will be different types of platelet coming from the young platelet and coming from the aged platelet. But also they have a different functional diversity for the platelets itself. For example, there's some subsets of platelet that for like the aggregations, and some subset for the coagulant, and some subset for the secretory, and some for the immune response. The reason why there's a different subsets here because the platelet actually is coming from the megakaryocyte in the bone marrow, and megakaryocyte there's a lot of different subsets of the megakaryocyte. Mainly there's three types of megakaryocytes in our bone marrow. So one is the inflammation, and the other one is for the aggregation. So that's why, and the other one is for the propagation. So that's why there's a different subtype of the platelets produced by the megakaryocyte. And also, in the disease condition, it depends on the different microenvironment. So they have different microenvironment, they will have a different stimulus, so they will leading to have a different types of the platelets. So that's why in both normal or healthy condition or in the disease conditions, they have different types of the platelets. So our group have been focused on working the role of the platelet in different disease. So what we found that is actually platelet have a very different forms of the platelet, and mitochondrial RS determine the phase of the platelets. So when we have activation for the platelets, actually they lead us some sort of the mitochondrial RS secrete from the mitochondria, and then they will leading to the activation of the platelet. But if the RS is increased gradually, for example, like in a mild situation, then they have a mitophagy that help to repair the mitochondria. And for the more severe situation, there's more RS, and they will become the apoptosis, and then they will leading to the platelet apoptosis, and they will secrete some of the apoptotic body, they will leading to more aggregations. But in more extreme or severe cases, they have a lot of RS accumulate in the platelets. They were leading to the so-called pyroptosis. They were leading to the explosion of the platelet, and then they will secrete a lot of the inflammatory cytokine. For example, like what we found is the IL-1 beta. They were leading to the cytokine storm in the sepsis. So based on all these findings, we also found that the activation of the platelet or the pyroptosis of the platelet can lead to the crosstalk of these platelets with the VSMC, monocyte, and the neutrophil. So what we found is actually the activation of platelets is not only they are for aggregation, for the blood clotting, but also they can interact with the smooth cell, and then they can regulate the function and the phenotypic switching of the smooth cell, and also they can interact with the monocyte, and then boosting them to the pro-inflammatory subtype, and also they can work with the neutrophil, and then leading to the neutrophil extracellular trap, the lactosis, and then leading to the more inflammations during the disease condition. So until very recently, the mitochondrial RS or the mitochondrial dysfunction is only the problem in the disease condition, but they are also a problem in the aging condition. So some of the recent paper has been found that actually in our aged platelets or in the old platelet, the mitochondrial dysfunction can leading to the different function of the platelet. For example, they will increase the degranulation of the platelet, and also they will increase the release of the inflammatory cytokine, so that make our vascular inflammations in the aging conditions. And also, this platelet can also interact with the monocyte, and then boosting them to the pro-inflammatory subtype. So that's why they will again leading to the more inflammations, and then and then leading to the thrombosis. But up to now, there's no answer for why there's a mitochondrial dysfunction in these platelets. Although there's some studies showing that maybe the inflammatory cycle, for example, like the TNF alpha itself, can impair the function of the platelets, but it's still is an open-end question. So we come to another concept, so-called clonal hematopoiesis. So actually, this clonal hematopoiesis is a process that's leading to the accumulation of the mutation in our hematopoietic stem cell. So there's some of the mutation that have already found in the clonal hematopoiesis process. For example, like the TAC2, DNMT3A. So these among all these mutations, so TAC2 is the one with the most significant effect to these hematopoietic stem cells. For example, for the hematopoietic stem cell, if they accumulate this TAC2 mutation, they will increase their cell renewal as well as they increase the competitive advantage. So that's why there are more and more these mutant stem cell in our bone marrow. And this mutant stem cell in the bone marrow will leading to the myeloid bias. So that's why there were more myeloid cell accumulate in our bone marrow, and then they will. And a recent paper has been shown that the macrophage that coming from this mutant TAC2 mutant hematopoietic stem cell will have increased risks of the fibrosis as well as the other cardiovascular disease, for example, like the aortic aneurysm. So that's why this mutation is very important to increase the risks for the cardiovascular disease. But the point is that under the myeloid progenitor cell, we have another cell called the megakaryocyte, which is the mother of the platelets. They are also coming from the myeloid progenitor cell. So if there's a mutation is because of this chromohematopoiesis, so what will happen to this megakaryocyte and what will happen to the platelets? So that's why we would like to know. So at the very beginning of our study, we're using the data from the UK Biobank, and then we do the analysis, and then we found that actually for those people that they carry this TAC2 mutation, they have a significant increase in the risks of the different thrombosis. So that's very significant compared to the other mutations in the group. For example, like the DMNT as well as the ASXL. So compared to the other mutations, the TAC2 have a significant increase the risks of the different thrombosis. And also, we want to know what is really happening in different ages, the parameters for the platelets. And then we found that for the platelet activation, they actually increase with the age. So we're looking at some of the very golden standard, for example, like the annexin 5, the CD62P, which is so-called the P-selectin, as well as the PAC1, which is the activation marker for the platelets. And we found that so along the different ages, for example, come to the 50 to 70, they have a significant increase compared to those under 30. So that means the platelet activation is increased with the ages. And also, we would like to see is there any changes in the mitochondrial functions. That's why we're looking at the TMRM, which is referred to the mitochondrial membrane potentials, and also the mitoSOCs, which refer to the superoxide coming from the mitochondria. And we found that actually, again, they are very quite similar to what we found in the platelet activation parameter. There's a significant increase in these aging groups. And then we try to link up the TAC2 mutation as well as the platelet activation marker, and then we found that actually the TAC2 expression is quite relevant to the. So they are inversely proportional. That means when there's a less TAC2, they will increase the platelet activations. And then we try to do the proof of concept experiment. So first of all, we try to lock out the TAC2 genes in the mice, and then we found that actually the loss of TAC2 in the mice can really promote the platelet activation and the thrombosis. So figure the panel A here is what we do for the looking at the time for the thrombosis, and then we found that actually for the TAC2 lockout mice, when we lock out the TAC2 in these mice, we found that they significantly shorten the time for the thrombosis. That means they speed up the process of the thrombosis compared to the fox mice. And then we're also looking at the parameters for the other for the platelets. For example, like as I said, we're looking at the P-selectin, which is the platelet activation marker, and also the other markers for the platelet activation. And then we found that actually for these mice and for these lockout mice, if we specifically locking out in the megakaryocyte in the platelet, and then we found that actually they have increased the activation of the platelet. And also we check the time for the first thrombosis as well as the other parameters that's showing the breathing time, the tail breathing time, and the occlusion time. We found that the loss of this TAC2 can significantly increase the time for the thrombosis and platelet activations. And then we also do the other two parameters about the platelet spreading and also the size of the clot. So it shows that actually the old mice with the knockout of these TAC2 genes in the platelet can increase the spreading of the platelet. That means they have more platelet activations, and also they increase the size of the clot. That means the platelet aggregation can also increase in this situation. And then because we think that the mitochondrial dysfunction is actually the reason why leading to the platelet activation in the aged subject. So that's we check the mitochondrial function in this TAC2 local platelet, and then we found that actually, if you can see from the mitochondrial membrane potential as well as the superoxide coming from the mitochondria, there's a significant increase in the old mice as we expected. And the knockout of this TAC2 gene in the platelet can significantly increase this TMRM, the membrane potential, as well as the superoxide. So that means these TAC2 are really affecting the mitochondrial function in the platelets. And also we are checking the morphology of the mitochondria. And also we would like to know whether this affect the redox balance, for example, the GSH, as well as the ratio of the GSH and GSSG in the platelet. And indeed, the TAC2 itself, by locking out this TAC2 in the platelet, we can find there's significant changes in these two parameters. So that means, in addition to the mitochondrial function, the TAC2 deficiencies can also leading to the redox imbalance in the platelets. And we really want to know the mitochondrial function, what's going on. So we did the Seahawk experiment that we measured the oxygen consumption rate as well as the glycolytic rate. And then we found that it's very interesting. We found that actually in the TAC2 deficiency platelets, the oxygen consumption rate is significantly impaired, but for the glycolytic rate, it is not affected. So that means the loss of TAC2 is mainly affecting the mitochondrial function itself rather than the glycolytic pathway. And then because what we're seeing is what we did is in the platelet, but we also want to know whether the lockout of this TAC2 will affect the function of the megakaryocyte itself. So that's why we try to use the CRISPR-Cas9 technology to locking out the TAC2 in the megakaryocytes, and we significantly do the lockout in the megakaryocytes. And then we try to, after we lock out the TAC2 in the megakaryocyte, we try to looking at the mitochondrial function as well as the other RS-related parameters. And then we found that by locking out the TAC2 in the megakaryocyte, the function, the mitochondrial membrane potential, as well as the other parameters like the RS and the mitochondrial RS, and also the superoxide dismutase activity, and the GSH and GSSG, those parameters for the redox balance in the megakaryocytes are significantly affected. So that means TAC2 deficiency is not only affect the platelet itself, they are affecting the megakaryocyte. And also, we do the oxygen consumption rate. We measure the oxygen consumption rate, and also we measure the glycolytic rate in this TAC2 lockout megakaryocyte. And then we also found that similar to what we've found in the platelets, the TAC2 lockout megakaryocyte can leading to the impair for the oxygen consumption rate, but a little effect in the glycolytic, in the glycolytic rate. And then we would like to know why the TAC2 can affect the the mitochondrial function, why they can affect the oxygen consumption rate in the mitochondria. So we did QTAC and whole genome bisulfate sequencing. We would like to know, because TAC2 itself is actually a methylation enzyme, methylase. So that's why we would like to know whether the TAC2 itself can do the methylation in this situation. So, and then we do these two techniques for the QTAC and the WGBS. which we would like to find is there any downstream target for the TAC2. And then we found that actually mTOR is a downstream target for the TAC2 in this way. And then we do the Western blot. We show that if we lock out the TAC2, it really affects the expression of the mTOR as well as the mTOR phosphorylations. And also the mTOR downstream signaling, for example, like the AKT, PSLU, as well as the P70. So those are the downstream signal for the mTOR, and then signaling component for the mTOR. And also we also found that the mitochondrial biogenesis parameter, for example, like the PGC1 alpha and TFAM, those are significantly changed in the TAC2 lockout mice. So that means at least they can provide some insight that the TAC2 lockout are actually affecting the mTOR signaling as well as the mitochondrial biogenesis. And also because we want to know more about how the TAC2 itself are affecting the energy metabolism in the platelet. So that's why we also do the metabolomics as well as the proteomics. And then we found that there's a lot of some of the very interesting metabolic pathway, for example, like the biosynthesis of the unsaturated fatty acids. As well, we found there's some changes of the metabolic pathway, and then also they leading to some of the very interesting process, for example, like the ferroptosis. But those analyses are still ongoing, and hopefully we can have generate more meaningful data in futures. And then very similar to what we did, we also do the proteomics study, and then we do the comparison, and then we found that there's a significant change in this TAC2 lockout mice. So that's why we would like to pursue and do the more in-depth analysis to see is there any more important metabolic pathway involved in this TAC2 knockout platelets. So to make the long story short, so what we found actually is in this in the aging situation, and we have accumulated this TAC2 mutation, and it will leading to the TAC2 deficiency or in terms of the expression and functions. So they will cause the downregulation of the mTOR, and then they will leading to the mitochondrial dysfunction as well as affecting the energy metabolism inside the macrokaryocyte. And then that's why they will generate some TAC2 deficiency phase, and this kind of platelet can also affected in terms of the mitochondrial function. So that's why this kind of platelet is not they are unstable compared to the young and healthy platelets, and then they are more easy to activate in the vascular and then leading to the thrombotic events. So in summary, so what we find is in the age-associated TAC2 mutation is associated with an increased risk of the thrombotic disease, and this age progressive platelet hyperreactivity is caused by the at least what we find is the mitochondria dysfunction and also the redox imbalance. And also the TAC2 deficiencies can promote the platelet activation and thrombosis by changing by impairing the mitochondrial function. And then what we believe our data have demonstrated that the TAC2, this gene, can at least safeguard the mitochondrial homeostasis through this mTOR signaling. So hopefully we can share more data in futures. So last but not the least, I would like to say thank you to my groups. They are all working on the platelet and vascular disease, and also my collaborators at Yale, and also my collaborator at Australia. So and also from the mainland China. And then so that's all for my presentation. Thank you.
Thank you, Dr. Tam. So any questions? Any questions? Yes.
So very interesting talk. Can I ask whether loss of TAC2 will affect the nuclear localized mitochondrial gene expression during the aging process?
Sorry?
Will loss of TAC2 will affect the nuclear localized mitochondrial gene expression or not?
Actually, they affect a lot of different genes. So at the very beginning, they found in this macrophage or super cells, they actually affecting the, for example, the KLF4, they leading to the super cell differentiations, and also in the macrophage, they found that they are affecting some of the inflammatory genes, but I don't exactly remember what it is. So that's why they found that the loss of the TAC2 will leading to the pro-inflammatory macrophage. and then leading to the inflammations. But I mean, there's not only one or two genes. There's a set of genes affected by the TAC2 because it is a methylase for doing the methylation in the DNA.
So I have actually kind of related question. I mean, we also published papers on TATs, mostly TAT3, but I mean the mechanism by which TATs regulate transcription. I still think it's a little bit enigmatic, whether this is primarily via affecting then demethylation, so methyl cytosine, or whether it acts as a per se label for readers. So what's your idea also related to mTOR signaling? I mean, how does it work that changes in hydroxymethylcytosine then affect gene expression and then downstream signaling? Do you have any idea how this works?
This is a very good question. So at this stage, we haven't know much about how the TAC2 is regulating in the macrocyte in the platelets. So what we've shown is just those phenotype data. So that's why we did a lot of the proteomics and the proteomics to see actually what is happening inside the megakaryocyte. Because megakaryocyte is very different from the other cell type. They have not only 2N, they have 4N, 8N, and even 16N. So that's why, I mean, the TAC2 maybe have a very important function to regulate this DNA.
May I know where is the TAT2 expressed in the platelets? Is it in the membrane? And the second question, is there any way to reactivate the TAT2 so that the platelet can be function well?
Yeah, that's a very very interesting question. So actually, TAC2 is located in the nucleus in the cell because they do the methylation of the chromosome. But in the platelet, we found that actually they can scatter in the cytosol. They are scattered in the cytosol, but we don't know what is the purpose of doing this. Maybe because the platelets are actually coming from the megakaryocyte, so that's why they have left some TAC2 there. But for our study, we just found that the TAC2 in the megakaryocyte can already leading to the decrease the expression of the mTOR and then leading to the mitochondrial dysfunction. And those mitochondria are actually going to the platelet. So this dysfunctional mitochondria will leading to the platelet hyperreactivity. So that's what we are proposing. Yeah.
Okay, thank you. Any further questions? No? So.
Okay, so we move on. Thank you very much. It's my pleasure to introduce the third speaker of the session, Professor Wei Kang. Professor Kang is the chair of the Department of Physiology and Pathophysiology at Beijing University Medical Sciences Center, where she also serves as the Dean of the School of Basic Medical Sciences. She's highly accomplished in vascular research with a focus on atherosclerosis research, which will be the topic of her talk. She'll be talking about novel players in atherosclerosis, cardiovascular disease. Looking forward to your talk.
Thank you. Thank you for the kind introduction. And I also would like to thank Cassie Liu and Zhou Bin for hosting this fantastic symposium. Actually, this is my second time in Hong Kong. Last time is 15 years ago, organized by another Cassie. So today I'm going to share about some of our study in atherosclerosis. Atherosclerosis-related cardiovascular disease, including myocardial infarctions, stroke, still accounts for more than 60% of morbidity and mortality all over the world. And currently, the major anti-atherosclerosis medicine is targeting LDL cholesterol. Back to 100 years ago, pathology from Russia tried to feed the rabbit with the sunflower oil and successfully induced the first animal model for hypercholesterolemia. And later on, the cause effect between hypercholesterolemia and atherosclerosis are well established. And later on, a number of breakthroughs has been made, including the HMG-CoA reductase identification, which is the key enzyme for synthesis of cholesterol, and LDL receptor, as well as PCSK9, etc. So you would appreciate that the current anti-atherosclerosis drug, including statin, which is the inhibitor for HMG-CoA reductase, and ezetimibe, which is the inhibitor of NPC1L1 to inhibit the intestinal absorption of cholesterol, and also the PCSK9, which will hydrolyze the LDL receptor and therefore inhibit the LDL cholesterol absorption. So all the drugs are targeting LDL cholesterol. They are very effective, actually, because about 1/3 of cardiovascular event can be inhibited through this pathway. However, even with statin, ezetimibe, or PCSK9 monoclonal antibodies, still there's more than half of the patients will suffer from the adverse cardiovascular event, even their cholesterol level are controlled well. And the thing's getting more worse if the patients suffered from other metabolic disease, for example, diabetes or chronic renal failure patients. They are more susceptible to atherosclerosis, and about 60% of patients with end-stage renal failure will die from cardiovascular or cerebral vascular disease. Unfortunately, the statin or ezetimibe, they're not working well for those end-stage renal failure patients. So our question is, what else can we do besides lowering LDL cholesterol? If we looking back, all the strategies are targeting controlling the risk factor. However, we do little things to target the vessel itself. Of course, there's some other ways we're trying to do because, for example, the Novartis are developing ASO for lowering LPA, which is a special LDL particle with apolipoprotein A. A number of clinical studies suggest it's an independent risk factor for cardiovascular disease. This is a phase 3 clinical trial. Hopefully this year we'll get some positive result. Another strategy we're trying for a long time is anti-inflammatory treatment. Although the interleukin-1 neutralizing antibody is showing some protective effect for atherosclerosis patients, however, they increased the infections risk. And interleukin-6 antibody, we don't know yet. And the only drug has been approved by FDA is colchicine, which is actually a very old drug. And it shows about 30% decrease for the cardiovascular event. However, there's some side effect, and also we don't know yet whether it's also protective for the further cardiovascular event. So still long way to go for this strategy. So how about vessel itself? Actually, yesterday, Nieyu and Cassie showed very nice data that this microenvironment actually could manipulate cell behavior. For example, the extracellular matrix back to 15 years ago, thanks for the progress for the proteomics technique, there's more than 150 matrix protein identified in human aorta, and more than half of them are proteoglycan and glycoproteins, and most of them, we don't know yet what's the function. It's just like Cassie yesterday mentioned, it's not just a scaffold, but also give fine-tuning the cell behavior. For example, manipulate the growth factor or cytokine activity, or through binding to different proteins like membrane receptors to adjust the signaling. And so the first question we would like to ask is, what is the important ECM protein to maintain the homeostasis of the vessel? One of our favorite molecules is called COMP. It's actually firstly identified in cartilage. It's cartilage oligomeric matrix protein. Its mutation could cause cytochondroplasia, which is the patients characterized by the warfarism and early onset of arthritis. However, the COMP protein is also abundantly expressed in vessels, and especially in smooth muscle cell. if we delete COMP, then the ApoE-knockout mice, even without high cholesterol diet, still are more susceptible to atherosclerosis, which means it's protective matrix protein. And how could this happen? We identified several mechanisms. For example, although endothelial cell does not express COMP, but if there's a COMP in the vessels, then the endothelial cell will be protected from auxiliary flow-induced inflammation through binding to integrin alpha-5 beta-1. And also, COMP is necessary for the smooth muscle cell to maintain the contractile phenotype. Without COMP, the smooth muscle cell will switch to other phenotype and therefore promote new intimal formation. And interestingly, COMP could even directly bind to thrombin and inhibit the further platelet activation and thrombosis formation. So, which means through modulating the whole vascular environment, COMP could protect the vessels and in response to some risk factors. So the next question is, what is if COMP is the protective factor, so what would destroy it? So by using COMP as a bait, we have been able to clone the metropolis called ADAMTS7, which belong to Adam's family, could degrade COMP. And later we identified that through degradation of COMP, ADAM7 could promote smooth muscle cell migration, and through degradation of another matrix protein called TSP1, ADAM7 would inhibit endothelial recovery. That's why if we delete ADAM7, we'll see dramatically decreased new intimal formation and as well as atherosclerosis plaque. And later, there's a number of studies identified that ADAM7 is actually a novel locus for human coronary atherosclerosis as well as stroke. In several years ago, we have been able to develop ADAM7 therapeutic vaccine. So the theory is like to promote B cell to produce neutralizing antibody against ADAM7. By application of ADAM7 therapeutic vaccine, we identified that it could successfully reduce atherosclerosis in mice, not only in ApoE but also LDL, not only for prevention but also for the treatment, without disturbing the cholesterol level and triglyceride level. We even tested this vaccine in the mini pig. We put the stent in the coronary artery of the mini pig, and to induce new intimal formation, we observed a similar protective effect for this vaccine. And more interestingly, we found this protective effect is very specific only for ADAM7, but not for other members of Adam's family, which means the strategy not just controlling the risk factor, but also targeting the matrix microenvironment, we can also get some beneficial effect for the atherosclerosis. And by using of TMPP N-terminal labeled proteomics, we also be able to identify novel substrate for ADAMTS7, which is complement factor H. It's actually inhibitor for overactivation of CYP3A and CYP3B. So therefore, which means ADAM7 inhibition could be effective for autoimmune disease. Actually, we tested in the acute renal injury model as well as the lupus nephritis model. It's really worked. So this is our paper published recently. We found that ADAM7, through degradation of COMP, as well as CFH, it can confer both protection, for both renal and vascular calcification. So which means we can find one target for both organs. And we also recently developed the humanized monoclonal antibody for potential translation. And for this story, we feel that this strategy might work if we could find more protective ECM protein. We could be able to protect the vessels more. So this is a very preliminary data for the human carotid enterectomy, and we perform the extracellular matrix proteomics, and you would appreciate just a different stage of this plaque. The microenvironment of the matrix is quite different. Hopefully, we'll find some exciting data later. And the second part of my talk is, in addition to extracellular matrix, actually, the vessels are facing a lot of risk factor injury. And for each risk factor, probably they have to play the role through some kind of receptors, channels, or membrane, some other membrane proteins. In addition to cholesterol and triglyceride, actually there's more lipid in our body. For example, phospholipid or sphingolipid. And for many years, sphingolipid and phospholipid, we are more focused on the membrane, cell membrane, or intracellular second messenger. However, they're not just intracellular molecules. For example, ceramide, which is one of the simplest sphingolipid, is composed of fatty acid and sphingosine, and according to the carbon number, and can be divided into long chain ceramide. For example, carbon 16-0, carbon 18-0, very long chain ceramide, and unsaturated long chain ceramide. For example, carbon 24-1. And back to 30 years ago, people already know ceramide is a component for lipid raft, and also it's very important for apoptosis, differentiation, etc. But recently, number of clinical study has identified that circulating long chain ceramide, including carbon 16-0, carbon 18-0, and carbon 24-1 ceramide, could predict cardiovascular death independent of cholesterol. And the Mayo Clinic has developed a CERT-2 score for the prediction of cardiovascular risk. And 2 years ago, the Science paper published a comment. They mentioned the 59 years old women who had actually normal biological parameter examination. However, her father, her sister, and herself suffered for the cardiovascular event. The only thing abnormal is increased level of circulating long chain ceramide. So our question, but so far although there's some compound has been developed preclinically, however, there's nothing has really go to the clinical study. So the major strategies like inhibit the de novo synthesis of ceramide or promote the clearance of ceramide. So our question is, if ceramide, circulating long chain ceramide, could predict a future cardiovascular event, whether it's a real cause effect for atherosclerosis, and if so, how circulating long chain ceramide could aggravate atherosclerosis. Because my lab also working on GPCR, which is a super family containing more than 800 family members, could almost sense everything. And about 36% of FDA-approved drugs are targeting G-protein coupled receptor, especially in cardiovascular field. For example, the ARB, the P2Y12 inhibitor, etc. So we are thinking if circulating long chain ceramide could affect atherosclerosis, what is the potential G-protein coupled GPCR. So we have to really figure out a readout for in vitro screening. So this is the strategy we're trying to identify the receptor. So we tested whether inflammation could mitigate this effect, because for a long time we have recognized atherosclerosis is a chronic inflammatory disease. And if we want to find a GPCR, we have to identify which G protein coupled GPCR it possibly be, because there's more than 800. It's really hard work to screen everyone. So by using of G protein specific inhibitors and combining with the single cell sequence data and the BRET assay, finally we have been able to figure out the real GPCR. The first test is we found that carbon-16 ceramide could induce both interleukin-1 production in both endothelial cell and macrophage. And in vivo study is suggesting that if we infuse the carbon-16 to the mice, it will dramatically increase the plaque without alter the cholesterol level and triglyceride level, which means, and if we deplete into caspase-1 or using the neutralizing antibody to block interleukin-1 or using the interleukin-1 receptor inhibitor, this aggravated effect will be abolished, which means the inflammasome could mitigate the long chain ceramide aggravate atherosclerosis. So we use this strategy for further screening, and we tested the second messenger, and we used different inhibitors targeting different G proteins, and we found it's GQ coupled GPCR. And here's the further screening by combining the single cell sequence data in both endothelial cell and macrophage. We have been able to screening 59 GPCRs, and fortunate we identified two potential receptors. One is CYSTR2 and the other one is P2RY6. The endogenous receptor for CYSTR2 is actually LTC4 and LTD4. The P2RY, the endogenous ligand of P2Y6 is UDP. So the IC50, the EC50 value for both receptors is around 0.3 micromolar or 2.6 micromolar. We tested the selectivity of the two receptors for different kind of ceramide, because ceramide is composed of fatty acid and sphenoxane. So free fatty acid and sphenoxane alone does not recognize these two receptors, and the carbon-16 could recognize both receptors, but carbon-18 0 could only recognize CYSTR2. So it's quite selective. So we also tested our strategy in vivo. So if we deplete CYSTR2 or P2RY6 or both knockout mice, then the atherosclerosis will be dramatically inhibited, and the cholesterol level circulating long chain ceramide that's not altered. And if there only, can we extend our study to the clinical application? We collect, because I mentioned for the CKD patient, they're more susceptible to atherosclerosis. However, the statin does not work very well. So we collected the blood from these coronary artery patients, and the circulating long chain ceramide level gradually increased with the decline of GFP, the renal function, with the decrease of the renal function, and circulating long chain ceramide positively correlate with the severity of coronary artery in patients. So we cut most of the 5/6 of the kidney from the mice to induce the chronic renal failure. And you would appreciate the third column. even the cholesterol level, triglycerol level remain same, the plaque dramatically increase. But if we deplete the CYSTR2, P2RY6, or both receptors, this will be dramatically reversed. So which means just by blocking the interaction between ceramide, circulating long chain ceramide, and the receptor in a vessel, we'll see some beneficial effect for atherosclerosis. By collaboration with Professor Jinpeng Sun, we also identified the Carol-EM structure for ceramide CYSTR2 GQ complex. So it's, I'm not kind of a structural biology person, but I would say this is a typical structure. There's two different domain. One is phenosine chain binding channel, and the other one is fatty acid binding pocket. Recently, we just screened some compounds, small compound, to targeting the CYSTR2. Hopefully, we'll get more positive data later. So, so the take-home message is like, for atherosclerosis, we think in addition to LDL cholesterol control strategy, we probably could do more things. One thing is to maintain the homeostasis of vascular microenvironment, for example, the extracellular matrix. The other thing, we probably need to figure out more risk factors and trying to find out the receptors to block the interaction. I would like to thank all my collaborators and thank you for your attention.
Thank you very much for this very concise presentation. We're a little over time, but we should allow ourselves time for one or two questions. Enzu.
Hi, Kui. Very nice talk. We know that in plaque, the contractile SMC will become synthetic SMC, and even they will become those macrophages. I wonder if the gene you knock out, like COMP or ADAM7, will have direct effect on the cell fate transition from the contractile SMC to those macrophage-like cell in the core of the plaque.
Actually, although I'm not the stem cell person, but we do publish a paper several years ago by collaboration with Qingbo. So with COMP itself could promote the stem cell differentiate towards smooth muscle cell. And COMP could also bind to integrin to manipulate the cell fate. And also we have another paper said COMP could bind to 81 receptor on the membrane. So it's, because it's quite big, so it can bind to a couple of proteins.
Okay, thank you.
A quick question. The long-chain ceramide, actually, you propose as a direct binding partner for the GQ protein to change the outcome in the downstream signaling. Who are the receptors?
Receptors.
Yes.
Is receptor, yeah.
GQ is actually the...
No, it's CYSTR2 could couple to different G proteins.
Through the GQ.
So it's only through GQ.
So the binding is still outside on the receptors.
It's on the membrane on the receptor, yeah. Thank you.
Hi, I learned a lot from your talk. Thank you.
Thank you.
I just wondered, because you didn't mention whether anybody has tried to use targeting inhibitors of calcification, like matrix glad protein, for example. Because I remember the paper that showed the knockout of MGP showed, hypercalcification of the arteries and then rupture. So, and also further interesting results showing that in the repair process, you actually form cartilage in the blood vessels. And so I just wonder whether people are thinking about using that as a target to inhibit calcification.
Yeah, it's a very good question. Actually, when we're getting old, we probably will get both vascular calcification as osteoporosis. So lost the calcium in the bone, but gain the calcium deposition in the vessels. However, all these drugs targeting osteoporosis does not show any beneficial effect for the vessels. So right now there's no drug at all. So I think they share some similar biological process. However, we don't know yet.
Short last question.
Thank you. It was a wonderful talk. Regarding your ADAM7 vaccine, I wonder when would you propose that people would actually be administered the vaccine if it was a therapeutic, and what are the downside to having a vaccine if ADAM7 has the maintenance function, or does it?
Yeah, that's a very good question. Actually, we asked this question with some famous cardiologists. Theoretically, because ADAM7 has been identified both genetically and for our biological study, so if you got this mutation, there's more susceptible. However, because ADAM7, as long as it increased, because it's generally is expressed as very low level during normal condition or physiological condition, so as long as it increased, it probably will give some effect. So we are right now developing some episode-specific antibody, trying to recognize if when ADAM7 will be increased. Theoretically, as long as the patient has some family history, some risk factor, we probably can apply this vaccine. It's not like one-time use, probably have to repeat it. But compared to neutralizing antibody, we can extend the timeline to like every 3 months or every half a year. Yeah.
Thank you so much again.
Thank you.
Okay, so may I invite the next speaker, Dr. Owen Go. So Dr. Go is a clinician scientist at CUHK. He holds a Bachelor of Medical Science and MBCHB from CUHK and PhD in Neuroscience in UCL. So he mainly studies in basic of sensory perceptions and the connection between the neuron. And he is investigating neural circuit and molecular mechanism underlying nervous system dysfunction in aging and neurodegenerations, and aim for identify new therapy. So welcome, Dr. Ko.
Thank you. Thank you very much, Ronald, for the kind introduction. I thank the organizer, Kathy, and also Professor Zhou Bin for giving me an opportunity to share some of our works we're doing at the Division of Neurology at the Chinese University of Hong Kong. Before I start, I'll be principally talk about a study we just recently published 2 months ago. And before I start, I have to acknowledge I'm really quite fortunate to be working with some of the brightest young scientists young biomedical scientists in Hong Kong, especially Dr. Junze Huang and Dr. Andrew Kwok. And also, I am also fortunate to be working with some brilliant clinical collaborators, so that some of the basic science stuff we can take to the clinics and test. And there are also 2 key contributors to the study I'll be sharing today, who are undergraduate medical students and co-first authors on the papers as well, who are Jason and Clement. So these are bright students who I've had the fortune of hosting at our lab. So the title of my talk is we're trying to devise sort of more anti-aging mechanistic driven treatment for cerebral vascular diseases. And the focus of why we're doing this is that we hope to have very pragmatic approaches that we can take to the clinic and test whether they can slow down the progression of age-related cerebral vascular disease. And I guess I don't have to convince the audience how important it is for us to tackle age-related disease given the global aging population. And our particular focus, we take a strong interest in age-related cerebral vascular diseases, especially cerebral small vessel disease and large vessel occlusion stroke. What is cerebral small vessel disease? It's the lesser known cause of dementia that catches less attention than Alzheimer's disease, but it's super important. Disease-wise, as its name implies, it's a disease of the little vessels of our brain. For example, in MRI, in T2 weighted MRI or DWI, in patients, what we typically see on its manifestation is that as disease progresses, there's more this whitening of white matter in the deep regions of the brain, and progressively that goes from, scatter little spots here and there, become confluent, merging together, and then become extremely extensive. And the underlying pathophysiology is extremely complicated. A lot of cellular and, molecular events occur or happen. That includes, for example, a bit of neuroinflammatory changes, breaking down of little vessels causing little micro spots of microbleeds here and there, and also occlusion of little vessels, and followed by secondary to vascular degeneration of the axonal chest that are mediating communications between brain region, therefore progressively leading to functional decline of the brain. And overall, apart from a small minority caused by strong genetic cases, it's principally a very strong manifestation of cerebral vascular aging. And why do you think it's important to tackle this? Is because compared to the Western population, the Caucasian population, this is actually an extra sort of prevalent disease or disorder in the Asian countries, especially in Chinese. For example, for those who are age 60 or above, almost 2/3 of us would harbor some form, at least mild form of cerebral small vessel disease, and a proportion of the patients suffer from moderate and severe cerebral small vessel disease. And cerebral small vessel disease is actually, first of all, the second commonest cause of dementia in the world, just after Alzheimer's dementia. And in fact, up to 80% of Alzheimer's dementia patients, when they are old enough, for example, when they're 80, for those who have Alzheimer's disease, 80% of them actually co-harbor vascular pathology or co-harbor cerebral small vessel disease. So it's a comorbid condition, most common comorbid condition in the brain that contributes to dementia. And secondly, it's also one of the commonest cause of stroke, and especially in globally, the figure is roughly 25%. So it contributes to 3 to 4 million of small vessel strokes per year in the world, whereas in the Chinese population, it's even more prevalent, accounting for almost, for example, this is 10-year data or 15-year data from Hong Kong, and it contributes to almost 40% of stroke in Hong Kong. So it's extra important for us to tackle. In terms of current medical treatment for this condition, we principally, as clinicians, we focus on disease risk factor control, but there isn't really anything that's genuinely disease cause modifying. But since I said that, it's a very strong manifestation of cerebral vascular aging, so it's important that ideally, we want to tackle the aging component, and also we want to identify molecular pathways that are on top of aging, contributing to the progression or a more severe manifestation of cerebral vessel disease. This is a very sort of active area of research. And for us, our starting point is focusing on aging first. So this is just to illustrate a lot of things happen in the aging brain. Basically, any cell type in terms of how they function, their molecular configuration, their cellular function, or any pairs of cell type in terms of how they interact are different in the aging brain. And it's important to consider whenever we think about aging, it's important to not just focus on aging of the brain itself, but also consider the aging of the whole organism, because obviously the brain is one of our body organs. It doesn't age alone. Whatever changes that happen is systemic conditions like circulation affects what the brain is exposed to, and therefore the brain, of course, is tied to the periphery. On the other hand, we neuroscientists, of course, like to think of the brain as the most important organ, comparable to the heart. In fact, of course, the brain, in fact, controls the body-wide neuroendocrine processes, right? So aging of the brain itself does have huge effect on the entire living organism. For example, 10 years ago, there was a couple of landmark papers from Cai Dongsen's lab demonstrating that by driving hypothalamic microinflammation just in the hypothalamus region on its own is sufficient to drive systemic manifestation of aging, and counteracting microinflammation in the hypothalamus is capable of delaying the appearance of aging phenotype body-wide. So for us, when we think about the problem of want to tackle cerebral small vessel disorder, we want to tackle that by targeting brain aging, then we should be thinking about the whole body or organism aging. Then, in order to do that, what kind of therapeutic modality or stuff we should try? Originally, we were thinking about this from a clinical angle, like what can we take practically to the clinic to test? Ideally, there are a couple of factors we want to be considering. For example, ideally, it should be a pharmacological approach, because compared to other more more experimental approaches like transfusing a, the young circulation, young plasma for a living organism, which is cool in science, in experimental science in animals. That's pretty cool, but then it's difficult to translate. And we also need to think about, there are also other means, for example, calorie restriction or time, restricted feeding. Those have very strong scientific evidence in terms of age counteracting effect. But then it's pretty hard to implement this in elderly. For example, can you please not eat for 16 hours a day? That's, I think, very hard to implement. So we want pharmacological approach. We need to be sure that the kind of approach we choose is safe. It has a safe, wide therapeutic window so that it's easier to attain sort of the a window whereby you don't have to worry about the overdosing, which is quite a challenge for example, rapamycin, the strongest known anti-aging drug, but suffers from side effect and narrower therapeutic window because at high doses is a very potent immunosuppressant, and it has other toxicities. And of course, we hope that the kind of experiments we want to do will be useful for guiding us in terms of what to pick and to test in clinical trial. And of course, while doing so, we want to understand the mechanisms. Since roughly 7-8 years ago, we started to work on GLP-1 axis. And of course, this is like a superstar therapeutic drug class and superstar therapeutic signaling. And the reason we started to work on this long ago while going from a cerebrovascular aging angle was that this drug class originally developed for the treatment of type 2 diabetes was found to be highly beneficial in diabetic patients, and that seemed to be, exhibit effects, a pleiotropic beneficial effect way beyond just glycemic control or even body weight control effects, right? So and at that time, even in 2017-18, there were already quite some evidence showing the promising potential of targeting GIP1 signaling in neurological disorders. For example, before us, there were already quite a few studies demonstrating that use of GIP1 receptor agonists can alleviate or slow the progression of disease phenotypes or changes in a couple of animal models of neurodegeneration based on familial genetic mutations. And then clinically, there were also evidence emerging, including, for example, in 2016. This is way before the recently announced EVOKE trial. This was in 2016. People already tested the use of liraglutide in Alzheimer's disease, noting that it could, in that trial, slow the decline of cerebral glucose uptake in Alzheimer's dementia. And the study that really promised to dig deep was this. This was actually published in Lancet by this exenatide trial in Parkinson's disease led by Thomas Fontini's team from University College London, showing that in idiopathic Parkinson's disease, use of exenatide was associated with slower decline of disease phenotype. And then last year, 2 years ago, there was another another study conducted by a French team showing consistent result testing this lisozanatide. So, but the study that really inspired us back then was this one. We were thinking that, in order to explain the kind of very wide-ranging effects of GIP1 signaling in this whole range of models, could it be that it was just a very potent anti-aging drug on its own? So initially we conducted some studies testing what happens if we just give naturally aging mice GIP1 receptor agonist, noting that in the first paper from my own lab, we're noting that in naturally aging mice, treatment with a GIP1 receptor agonist we were able to reduce the kind of age-related non-specific bubbling bear leakage by a lot. So this is if we quantify the extravasation of a small, medium-sized molecular type from the intracellular space to extracellular. In young animal, you don't see that kind of leakage. In aged animal, you see a lot of that. And if we treated the mice with an exenatide for just 1 month, it was sufficient to reduce that by more than half in this particular assay. And when we plotted the kind of transcriptomic effects in endothelial cells, comparing on the x-axis the aging effect, and y-axis the treatment effect, and each of these dots represent one gene, then we saw a global reversal of the age-related expression changes at the transcriptomic level. And then we did some further extension showing that this kind of effect, this negative correlation between aging effect and treatment effect also generalized very well to a broad range of glial cells in the brain. So it seems that it is a pretty potent, at least in the using transcriptomic readout, pretty potent drug that could reverse a lot of the age-related expression changes we see in the aging vasculature. And then we decided to do a full-scale sort of anti-aging study, and that was led by Junze Huang and later Dr. Andrew Guo, a clinician scientist, a brilliant guy from our own medical school, joined this project after graduating from Oxford. So we conducted these experiments whereby, first of all, we took some middle-aged animals who were aging, of course, and treated them with a given receptor agonist for a bit more than 6 months, and another group with a PBS, and also we include a young age animal for comparison, and conduct longitudinal assessment of a whole range of things, including a body weight, food intake, physical measures, cognitive tests, all that stuff. And by the end of the experiment, we collected body-wide tissue organs for multi-omic assessment. And this is what we found. Basically, in 4-limb growth strength, comparing the drug-treated animals to their counterparts of the same age but treated with a vehicle for the same duration, we saw a progressive improvement in 4-limb growth strength. So that's an indicator of muscle power. And we also see a consistent improvement in rotor rod tests. So that means that they are better at performing motor coordination. We also did some spatial memory tests, which I'm not showing here, but it did not attain statistical significance. But on some metrics, the drug-treated animals were basically performing in between the aged animals and the young animals. And because, like I said in the beginning, calorie restriction or time-restricted feeding or reducing food intake basically has an effect, anti-aging effect. We wanted to rule out the confounding effect from that. So before these experiments, we actually did some dose titration. We used a low dosage, which is almost body weight neutral and did not have an impact in glucose, so that we can largely attribute the kind of biological effects we've seen to the drug actions rather than confounded by body weight changes. And then, as I said, we collected body-wide organ tissues for multi-omic assessment, and they included transcriptome, of course. So I'll be showing a lot of scatter plots. We had a few variety of plots, but the scatter plot is the most straightforward for illustrating effect. So basically, with this scatter plot, on x-axis we're showing the aging effect. On x-axis we are showing the treatment effect. Imagine if the treatment had nothing to do with aging effect, then we would be expecting the equal number of differentially expressed genes to be falling in the second and third quadrant versus the second and fourth quadrant. But universally, across body-wide tissue organs, we saw almost universal sort of anti-correlation between aging effect and treatment effect, and that also generalized beyond transcriptome to circulating metabolome. And of course, we did some pathway enrichment analysis showing that for those the gene sets that are functionally important and related to aging, defined as hallmarks of aging and so on, basically after treatment, we almost universally saw a counteracting effect with drug compared to aging effect. And apart from transcriptome, circulating metabolome, because epigenetic changes is also considered by the field as an important contributor to aging, and apart from being a manifestation, we also did DNA bit array-based methylation assay using the 320K customized array from Steve Hovath's lab, developed in collaboration with Illumina. And again, we saw this kind of global counteraction in many tissue organs, and that was especially strong in 4 tissue organs, including the hypothalamus, frontal cortex, so 2 brain regions, and also includes the heart and adipose tissue. So it seems that we could see this kind of anti-correlation between treatment effect and aging effect across multi-omic and across multiple tissue organs across the body. And then for aging, when we try to claim something is anti-aging, there are different possible ways it could work. For example, one possible way is that it just so happens that all the molecular effects have the exact opposite direction compared to aging, but then it has nothing to do with aging. Under that model, we will be expecting to see the same kind of molecular impact or functional impacts in young animals. Therefore, we did an additional sort of parallel batch of experiment and show that we did not see that in young animal, but rather the kind of functional benefits and molecular impacts were quite specific to aging animals. Therefore, more supporting that the drug treatment was really trying to modulate or slow the aging process rather than having a non-specific effect that just so happened to be opposite of aging. So this is what we did. And in addition to that, we also dig a bit deeper here to understand the mechanism a bit more further, because this drug has multiple potential target sites in the body. Originally developed for treatment of type 2 diabetes, it's highly expressed in pancreatic beta islet cells. That is for potentiating insulin release when there's hyperglycemia. In the brain, it's actually a very interesting neuropeptide because there's a almost parallel axis of signaling produced by a specific subset of neurons in the brainstem solitary tract nucleus. These neurons, they have long-range projections to the hypothalamus, co-releasing glutamate and GLP-1 there in order to modulate downstream processes. So we wanted to narrow down further the receptor dependence or mechanism, and also partly inspired by others' work, for example, Cai Dongsen's work showing the importance of hypothalamic microinflammation in aging, and also Daniel Drucker, the GIP1 godfather's work on showing the importance of CNS GLP-1 receptor in mediating body-wide anti-inflammatory effects. We did these experiments whereby we just took an batch of even older animals, first of all validating that the kind of multi-omic effects are there, including across the transcriptome, across the DNA methylome, and a bit conserved still in the circulating metabolome. And interestingly, we observed that in terms of the overall strength of anti-correlation between drug effect and treatment effect, in this even older batch of animals, it seems to be even stronger. So perhaps compared to the previous batch of experiments where we used middle-aged to age mice, maybe simply because the aging has happened to a greater extent. Therefore, the drug anti-aging effect is also more obvious. And then we did this experiment whereby we specifically knocked down the GLP-1 receptor in the hypothalamus, and then compare the treatment effect again. And very interestingly, the kind of universal anti-correlation after knocking down hypothalamic receptor was almost completely gone in essentially all tissue organs and across multi-omic, with the exception of the hippocampus. We still haven't figured out why. But in other tissue organs, including the heart, for example, in the aged animals' hearts treated with a GIP1 receptor agonist, we saw a strong anti-correlation between transcriptomic impact of drug treatment versus aging, and also that also was happening in the DNA methylation level across hundreds of thousands of CPG sites. But then after knocking down the receptor in the hypothalamus, this kind of anti-aging effect in the heart was also completely gone. So for us, that's very interesting. Basically, the drug needed to act in the brain in order to attain anti-aging effect elsewhere, including in the heart. And then apart from that, we also did some benchmarking experiment because, like I said, mTOR inhibitor rapamycin is still the strongest known anti-aging drug, right? So we benchmark, in a way, GIP1 receptor agonist effect against mTOR inhibitor. First of all, mTOR inhibitor really lived up to its expectation in our lab. Its anti-aging effect was like super strong. You see this kind of anti-correlation across multi-omics everywhere. And secondly, when we compare the molecular impacts in transcriptome and in DNA methylome and circular metabolome due to GIP1 receptor agonist treatment and compare that to mTOR inhibitor, we saw them to be strikingly similar. So scientifically, we think this is very interesting because the particular GIP1 receptor agonist we used was exenatide, the first FDA-approved peptide Gib-1 receptor agonist. It actually came from the venom of Gila monster, so it's actually naturally derived, a natural substance. Whereas rapamycin, if we look back in history, it was isolated from a particular bacterial strain from Easter Island. So who would have thought that, by putting a little, a peptide from the venom of a Gila monster and, something produced in nature by a bacterial strain, they would have this kind of strong convergence of molecular impact across multi-omics and across the body. And this brings out, these findings brings out a lot of additional questions to us, which we are further investigating. But in particular, going back to why we did all these studies, is that we are interested in knowing what we can use to achieve anti-aging effect, in particular use that to treat cerebrovascular disorders. So in the field, this editorial came out just 1 week before our paper was published. Actually, the field also started to, there's also increasing voice in the field over the past few years, principally based on clinical observation, starting to consider GYLP-1 class of drugs might be anti-aging drugs. And our study, after we published it, was highlighted by Nature Aging. Recently, it's been announced that in the US, there will be quite a few mega trials starting to test even these ideas in human beings, prescribing, for example, semaglutide in healthy patients who are 60s to determine whether they have anti-aging effect. I think these are exciting developments. It would be nice to know, whether we whether the stuff that we first demonstrate in mice generalized to human beings. That's cool. But on the other hand, we take a different direction in terms of clinical validation. So trying to show that something is anti-aging in perfectly healthy patient requires large sample size, huge amount of resources, and very long follow-up. But then we want to do it a bit differently, and this is the guiding principle of our clinical trial. We want to pick diseases that have a early diagnosable, unambiguously diagnosable stage, and after which functional decline begins and accelerates, and tackle diseases that are in particular in the phase where there's a viable treatment window. And we think that we have to pick the right window because we were too late. Things, aging has occurred too much. A lot of degeneration have already happened. It's probably already impossible to reverse. If it's too early, just like the other trial that people are running, that would take huge resources, too many people, too much money. And we want to pick diseases that are strong manifestations of aging of the brain, and pick the right window to give the drug so that we can, we have the best probability of attaining a cumulative treatment benefit during the limited sample size, limited trial period during which we can follow up for the patients. And that was the basis on which we launched this trial, which we call the GAP-SVD trial, GLP-1 analog in the prevention of progression of cerebral small vessel disease, in which we pick specifically patients or subjects who are largely functionally independent, but they are already harboring moderate to severe cerebral small vessel disease in the brain. And so we started this trial long ago, like in 2021. It's still ongoing. We recently expanded it to a multi-site trial, including involving a mainland partner as well, because running a trial in Hong Kong has honestly been quite a pain in the neck. We simply don't have the patient volume, so sufficient patient load to support that. So we just extended to Chinese mainland, and we hope that by early 2028, this will finally be done. And before I finish, I also want to just highlight another related trial. So this is strictly speaking not leveraging the cerebrovascular aging aspect, but leveraging the anti-neuroinflammatory effects given receptor agonist led by Dr. Bonaventure Yip, another clinician neuroscientist of our neurological division, in which we pick large vessel occlusion stroke patients who are moderate to severe in terms of their functional deficit and severity of stroke. And this time, compared or different to many stroke trials run in the past, instead of picking patients and expect to try to see treatment effects just by prescribing a neuroprotective agent, we specifically choose patients who receive endovascular thrombectomy. And the theory being, we think that in these patients who have received endovascular thrombectomy, recannulization gives an opportunity for the drug if it has to act on the brain to reach the target site. And secondly, a drug on its own in large vessel occlusion stroke might not be strong enough to exhibit a beneficial effect. But then in the context of reperfusion, it creates a different biological avenue for anti-neuroinflammatory drugs to exercise its effect, be it anti-reperfusion injury or anti-neuroinflammation. And to our excitement, we observed that in a subset of patients who typically present between 4.5 hours to 12 hours of stroke, receiving just 2 doses of given receptor agonist would result in a probability of remaining functionally independent on day 90 after stroke by more than 50%. That is going from 40-something percent to over 67%. And we have recently completed a follow-up phase 3 trial already, and that the result of which will be announced by Bonaventure and our collaborator in the European Stroke Organization conference in May. And so that's it. So to quickly summarize it, of course, we know that aging is strongly related to cerebral small vessel disease, stroke. Our main contribution was experimentally we connected and demonstrated the mechanism, hypothalamic dependent mechanism or the brain-body axis mechanism of GLP-1's signaling effect to aging, and so that gives us some new mechanistic understanding. And secondly, on clinical studies, we are attempting to expand based on our mechanistic and also understanding and also experimental evidence its indication and application in neurological diseases, hoping that in the future patients with cerebrovascular diseases will have a better prognosis of outcome. In background, we are digging deeper in the mechanism, and we are also trying to develop new what we call multi-target engaging therapeutics, because having something to counteract the aging component is important, is a great starting point. But then what do we complement to it to achieve the best possible probability of attaining a real treatment benefit in patients? This is something we're working in the lab further. So that's it. And of course, I have to thank again all my team members, my collaborators, and also my funding bodies for making the work possible. Thank you all very much.
So thank you, Owen. So any questions? Yes.
Hi, Owen. Thanks for sharing this beautiful work. For the GLP-1 treatment, it can have good beneficial effect on those aging, especially neuronal phenotype. I'm wondering, at the cellular mechanism, if this GLP-1 treatment could have some effect on the senescent cells in the aged mice, or could they reduce the senescent cell number or reverse the senescent cells by some gene expression or function?
Yes, that's a very good question. So in one of the slides I've shown, we did look at we will pick so there are many these gene panels, gene sets, for example KEGG, GO, others, right? We pick these gene sets and look at these genes, senescence associated genes, sometimes they have different patterns of expression changes in different tissue organs. And then so we plotted the expression changes across different organ tissues versus in aging versus treatment effect. So it was a very strong anti-effect. So basically senescence associated. Whereas whether it reduces the number of senescent cells, we don't know yet. In one of our follow-up studies, we are doing single-cell transcriptomics and also spatial transcriptomics to look into, to compare the effects of GIP1 and mTOR inhibitor in the brain, because we want to understand the fine brain region and cell type specificity of these different types of anti-aging treatments. And one of the panels we are planning to investigate is exactly senescence markers.
Okay.
Yeah. So thank you so much for your question. That's a very good one.
Thank you. It's extremely impressive, and especially when you look at the concordance of the GLP-1 molecular signature versus mTOR inhibitor. But I wonder whether this is all because of the impact on the food intake, because that's also as a starvation signal that goes through the mTOR, or it's actually direct impact of the GLP-1 receptor mediated signaling.
Before we did these experiments, we did those titration experiments. We tested, so we use exenatide, and we tested 5 nanomole per kilogram body weight per day versus 10 nanomole per kilogram body weight per day for, dosing for the mice. And we chose the lower one because at 10, we started to see more obvious body weight reduction and food intake reduction effects. But with 5, we didn't really see that in the aged animals. So we were trying to rule out this potential confounding effect. Yeah. But because we picked a lower dose, and which was almost body weight neutral, we could be reasonably confident we could attribute that to the other pharmacological impact rather than body weight or food intake effect. Thank you. That's a very important question for related to interpretation. Thank you.
So often, do you think GLP-1 receptor will be helpful to prevent Alzheimer's disease?
Yeah, that's an extremely good question. So if you guys have been following up on Alzheimer's trial, recently Novo Nordisk since 2021 or 2022, they launched 2 large-scale phase 3 trials testing GLP-1 receptor agonists in Alzheimer's disease. And in the original design, they say they wanted to include 20% of patients with cerebrovascular comorbidity, and they also cited our paper. But eventually, somehow, they decided to go forward with almost purely Alzheimer's disease, and they picked early symptomatic stage. And the paper just came out actually yesterday in Lancet. If we look into the paper, basically, they did not see any benefit in clinical outcomes, but in cerebrospinal fluid neurodegeneration markers, there was a consistent reduction due to GLP-1 receptor agonist treatment. So for treating Alzheimer's disease at early stage, but already symptomatic stage, it seems that their trial showed that it may not work. But with generally, what we think, or what I think the field should move forward is going even earlier. For example, going to pre-symptomatic stage guided by biomarker, strong biomarker that is highly predictive of syndromal conversion, let's say in 3 to 5 years, and then treat them. Then maybe that will work. And another very important factor is that Novo Nordisk decided to go for oral formulation of their GLP-1 receptor agonists. But if we look at the pharmacokinetic studies that they have done and other teams have done, the variability of drug exposure is much larger compared to subcutaneous injection. From my perspective, I think for extremely difficult to tackle clinical condition, it's important to minimize variability from other factors first. So I think we should go for injectable and we should go for pre-symptomatic stage. That's my take. Thank you.
Okay, thank you. Any further questions? No? Okay, thank you.
Thank you
very much. So may I introduce the next speaker, Dr. Ryo Sujimura. So he is currently an assistant professor in Hong Kong U Medicine and also principal investigator of the Center of Translation Stem Cell Biology, director of the Blood Engineering Laboratory. So he is mainly focused on studying the structure and induction strategy of CAR in immune cell from human iPS cells, and also human immune cell development and role of the checkpoints in using the single-cell multi-omic approach. So his talk is talking about vascular organoid studying the tumor microenvironment. Welcome.
Yeah, thank you very much. All right, so starting this, nearly I'm a lunch broker and I'm a worker. So we'll be trying to be like precise and on time. So modeling like tumor microenvironment using organoid, especially assemblyoid. All right, so here is a very famous, Ira Merman's picture, cancer immunocycle. In order for immune system to successfully treat cancer, you need an amplification of immune signals in both local cancer and remote lymph node. But actually, those microenvironment not only support immune system, but major case, they are preventing your particularly T cell. So tumor microenvironment is a kind of major blocker of your immunotherapy. Why? So if you go look at this inside of tumor, tumor grow like a baby. Why? Because they are basically hijacking embryo program, like CMYC, NIN28, bunch of famous embryonic genes are up again in tumor. But not only cancer cell itself, but their surrounding microenvironment also being back to be like embryo-like, which is called oncofetal niche. Especially famous characters are like endothelial cells, macrophage, and fibroblast. So essentially in the tumor, those players are being back to be like a baby and supporting cancer growth and suppressing function of T cell. So how to model this? All right. So here, okay. If you have a PDO, patient-derived organoid, you have some like a major issue, like after a bunch of passage culture, these guys, you are losing most of those guys, okay, immunocells, fibroblasts, vasculatures are gone, and you end up 100% in epithelial cells, or especially cancer cells. So Alex, in my laboratory, asked this question: can we supply back these guys, and then make your cancer organoid complete, having TME, tumor microenvironment? So how to do this? So we are actually more embryo biology, developmental biology guys. So we use iPSC, stem cell embryo model, trying to understand how human embryo develops and what is a precise consequence of disease, like congenital disorders. So this is actually our identity. But understanding those key mechanisms of human embryos, we know like those major players, as I said, EC, macrophage, fibroblast are coming from epiblast, primitive streak, advanced mesoderm. So it's basically mesodermal guys. When people making iPSC to differentiation, in vitro differentiation, we basically try to do like purification. Like each step flow cytometry purification to make EC, macrophage, or a fibroblast or mesenchymal cells. So that's what we do. Also, I have done this for more than 10 years back in my postdoc era. But Alex, what he did was, can you go all in one in 3D culture if you do all in one? Because essentially these are same process, same mesodermal population, hematoendothelial induction. You get all in one, like these components, EC, macrophage, and fibroblast. So this is his product. iPSC 3D culture and step-by-step of morphology in cytokine exposure. You have blood vessels, monocyte, macrophage, and those pillars are fibroblasts, which we call vascular immunoorganoid, VIO. So here is a section and morphology of this VIO, actually churning out blood. This is cut section, CD45 really rich, and you can see CD31 blood vessels, and they are actually going into EHT, endothelial to hematopoietic transition. Like this is a very familiar concept with hematologists like me. So having this system and how to use this one. All right. So that's more like a detailed structure of this VIO, and we can see stromas, and they are making blood and releasing out. Composition-wise, like 80% are stroma, 10 are immunocells, CD45, and then 5% are vasculature. And those guys came out are basically all CD45 positive. Then Alex have done single cell analysis, and they basically identify their components and bunch of monocytes, granulocytes, hematopoietic progenitors, fibroblasts. And what's important is actually here if you compare these guys with like human atlas, like real human cell types, like in embryo, fetal stage, they really correlate well. So that's basically indicating what we are getting in this VIO is very close to what we have in human embryo, or like a fetal stage of macrophage, EC, and fibroblast. So that's a bunch of those like metrics are showing like their correlation. And if you see their subsets, macrophage, you see famous like TREM2 or FOLR2, FABP5, basically 5, 6 populations. And fibroblasts also very heterogeneous. Some of them are making more ECM, and some of them are more dividing. That is basically corresponds to what people know in tumor microenvironmental macrophage and fibroblast. So this is what we have in VIO. So how to put back these guys into our PDO. So this is an interesting point. Okay. So very simple, very brief, and basically low technique and low budget. So we have any PDO like here, and you have a VIO. And this is 96 where you bottom non-adherent plate, you put one by one. So this is a VIO and your PDO like a set. And then you kind of tilt 45 degrees. So that's actually your plate tilting. And then 1 to 2 days, you can see like 90% of them are actually being fused. So that's a picture. We have a bunch of collaboration, like including HTU, UK, US, and also Australia, and got a collection of cancer organoid from patients. Colon cancer, lung cancer, GBM. And you can basically make these guys as a fusion with our VIO. So this is how they look like. All right, so cutting section, because that's a fun time to see how these guys are emerging. So example in GBM assembly order, we call cancer, vascular, immunized, cyber, a kind of bunch of long name. But in this case, you see VIO and PDO, and then this HE section are kind of showing like the insides contents are like flagging each other. And if you see more like in this 3D current 3D image, that's CD35, like those blood vessels are going into cancer site, and CD45 immunocells, essentially monocytes, macrophage, are also going to cancer. Cutting section much clearer, so this is again GBM, and you can see this PDO site having a lot of CD45 indicating monocytes, macrophage, and then PDG receptor alpha is a kind of stromal components. Also, they are going into this area where those two bodies are merging. Same case in liver cancer assembly oid, we can see CD45, also CD68 macrophage marker are more into GFP positive cancer area, and then same into colon cancer, and then like you can see a bunch of macrophage are going to those cancer because of chemokine. All right, so that's basically we can immunize and vascularize. Then a lot of analysis Alex has done because he loves omics, he loves computer analysis. So this is the case. We have the single cell analysis, and we compared our profile of example here, CAF, cancer-associated fibroblast, our assemblyoid versus primary tumor samples. In this case, LUAD, lung cancer, lung adenocarcinoma. We can see like CD44, periostin, like a very famous oncofetal CAF marker, consistently expressing our assemblyoid and also LUAD, primary patient samples. Then he cut a section, that's 10x VISIUM HB, like a slightly higher resolution version of VISIUM. And we can see nearly sub-single cell, nearly single cell level of interaction. So what he showed here was CAF, again CAF, and you can see like a bunch of those orange, yellow area is those CAF is depositing collagen, and in this area, like example, blue thing, you see endothelial cells and macrophage also forming cluster. That is basically spatial organization of tumor microenvironment you can see in those spots. And what's particularly happening here is actually those CAF is producing chemokines, CCL20 and CXCL40. Those are known to recruit macrophage. So then we check macrophage because they are very close to each other. So here is a population of macrophage, bunch of subpopulation. But the biggest change here we can see is IL-1 beta positive macrophage. So what this says. So this is a VIO. This is a macrophage after assemblyoid. So that means macrophage after encounter cancer. The major population you see here, a lot of these red guys are IL-1 beta positive macrophage. So why this matters? Because IL-1 beta, they're interacting with tumor expressing IL-1 receptors. Also we can see by this spatial interaction of IL-1 beta macrophage and receptor positive macrophage. So IL-1 beta, it's inflammatory. So maybe you might be like guessing, oh, that's an inflammatory macrophage, which is maybe M1, good macrophage for anti-cancer, but actually no, it's completely opposite. IL-1 beta is one of the most problematic pathologic inflammatory cytokine whose function is promote cancer metastasis. and suppress T cell. And this is exactly we see in our tumor-associated macrophage. They are rich with IL-1 beta. So what's this consequence? So we test the function. So having those bunch of IL-1 beta tumor-associated macrophage in our assemblyoid, we hypothesize maybe this assemblyoid is a very toxic environment for T cells, as IL-1 beta does to T cell suppression. So here is a co-culture experiment with our assemblyoid with allogenic aerosol T cells. And you see those T cells got expressed LAG1, PD1, exhaustion marker. And this allo T cell is usually very abrasive. They can eliminate PDO if you just co-culture within a few days. But in this assemblyoid environment, those exhausted T cells are not able to eradicate those cancer cells. So this data shows here T cells, and this is without T cells, say just assemblyoid. Their composition, cellular components are no change. That means their T cells become functionless, exhausted by potentially this IL-1 beta high environment. What IL-1 beta does here is also enhance metastasis via angiogenesis. So recruiting blood vessel and then promoting cancer metastasis. So we have checked what is a case in our blood vessel. Also, this is an image. Vasculature become really like a fenestrated, potentially permissible, like this we can see in this structure, sinusoidal endothelial cells in our assemblyoid. This is going into also cancer side a lot, like penetrating tumor side. Indeed, they are basically having some rumen, like dextran dye thing. We can see those vasculature go into cancer. This is basically HIF1 positive hypoxic region, and angiogenesis is going to the sites. What happens here is basically you can see this rumen section that cancer cell is kind of riding macrophage and doing intravasation. So go inside of those blood vessel rumen and trying to do a metastasis. All right, so this is near the final point. So far we have basically assemblyoid. We are able to supply macrophage, blood vessel, and fibroblast as if like a very oncofetal niche, which support cancer growth and suppress T cells. So what we can do with this system platform? So one thing is precision medicine. Like example, KRAS inhibitor is a major advance in precision medicine. Originally, that was a very first generation, like pan-RAS inhibitor, but becoming like a KRAS specific. Nowadays, we have a very precise mutation-specific KRAS inhibitor, like example, G12C KRAS thing. Because KRAS is a major driver of lung cancer, and we are very happy, lucky to have lung cancer PDO in QMH, basically those bunch of patients. We have a records of each patient, either response to KRAS inhibitor or not. And then actually our collaborator here, David Ramsey in HTU and QMH, they have those bunch of record and PDO bank. But what happens in our case was the PDO itself cannot recapturate the response in the patients. Even though we know like this drug worked in patient A, patient B didn't respond. But in either case, the organoid PDO shows they are just modestly responding to this chemical, KRAS inhibitor. So we wonder why, and we ask whether supplying back those tumor microenvironment will be able to rescue that response in patients. So this is an experiment we have done. We have done assemblyoid, and particularly the patients who just like modestly show response in PDO was able to show a better response if you supply back tumor microenvironment, as if it happened in the actual patient's record. So here is a summary of this talk. We are able to endow like stroma, vascular, and immune component into any type of PDO. We recapturate example IL-1 beta high tumor microenvironment, and then T cell exhaustion. we are able to test, like example, KRAS inhibitor assessment in more precise manner. I would like to thanks to example, this is done by Alex in my laboratory, and David in HKU, and Sharma, Australia, Vivian in UK, and Olivia in UCSF, and also funding source.
Thank you, Dr. Sashimura. So because of the time, probably we save the Q&A session later. Yeah. So we need to move on to the last speaker.
Thank you very much, and it gives me pleasure to introduce my co-chair, Professor Ronald Wang, here from Hong Kong. He's a professor and deputy director of Prenatal Genetic Diagnosis Center and a division head in the Department of Obstetrics and Gynecology here in Hong Kong. And we will look very much on your presentation on the pharmacogenomics and pharmacokinetics of aspirin in preeclampsia prevention.
Thank you, Chairman. So I want to keep it short because I also running out of time. I need to rush to the train stations for my next event. So my talk changed a little bit about the organs. So this is placenta, probably everyone know. And actually, there's a lot of vessels developed in the placenta. So here's the vasculature of the placenta. So they have a umbilical vein and also umbilical artery as well. So currently there's many pregnancy complications actually developed from the placenta. That's also relies on the vasculature of the placenta. And also currently we try to use the different methods, different medication, try to improve the circulations of the placenta vessels. So that's a difficulty for us to control the circulations in placenta. So one of the major diseases I would like to talk about is preeclampsia. So preeclampsia actually is a de novo hypertension developed after 20 weeks of gestation. So this is a new development of hypertension, so excluding those pre-existing hypertensions before the pregnancy. And they also complicate with the proteinuria and also many medical and maternal complications and also different end organ dysfunctions. So the major issues of the preeclampsia is there's a lot of maternal mortality and also mortality developed from this disease. Not just from the mother, but also from the baby. So once the mother develops preeclampsia, there's a risk of prenatal mortality and also morbidity as well. And also after the pregnancy, both mother and the baby will develop hypertension risk after a few years later. So this is alarm of a lot of attentions try to improve the pregnancy outcome to prevent the future development of cardiovascular diseases. So here's also showing the mortality of both mother and the fetus if the pregnancy complicated with preeclampsia. So right now, what kind of clinical methods try to prevent the development of preeclampsia? So the first, back to the 2 decades, probably different people using different methods to prevent the diseases, including pharmacological methods and non-pharmacological methods. So one of the pharmacological methods is heparin and also aspirin, but you can see the efficacy is quite low. And also non-pharmacological method is antioxidants and also different supplements, including calcium and also vitamin D. But you can see most of the time in the old trial, so showing that non-pharmaceutical method actually is more effective than the pharmaceutical methods. The reason of the failure of the pharmacological method is due to the study design. So I'm going to talk about. So until recently, one of my colleagues in my department, Professor Leona Poon, and start a new trial using the aspirin, starting from early pregnancy, around 16 weeks of gestations. And she randomized the patients into aspirin group and the placebo group. And in her study showing that aspirin actually can significantly lower the chance of the preeclampsia particular for late gestations. The efficacy rate probably around the 60 to 70% depends on the compliance of the low dose aspirin intake. But a striking finding actually is for preventing preeclampsia developed in very early stage of gestations. So we divide the preeclampsia, term preeclampsia, preterm preeclampsia as well. Even early onset of the preeclampsia starting from 16 weeks of gestations. You can see the efficacy of the aspirin can be really improve the prevention efficacy up to 80%. So after this trial, there's many countries follow this methods to start the low dose aspirin early gestation and try to prevent preeclampsia. Here's a meta-analysis report, and different country and different international guidelines change the policy to start the treatment early on. So overall, based on this meta-analysis, there's a consensus to show low-dose aspirin can prevent early onset preeclampsia quite efficiently and try to avoid the severe complications in preeclampsia as well. And not just the mother suffer from preeclampsia can be prevented, but for the complications of the baby also significantly reduced, including fetal growth restrictions and also prenatal mortality as well. So based on this meta-analysis, they performed a subgroup analysis of what kind of patients can get more beneficials of this low-dose aspirin. So one of the conclusions is we have to start the aspirin earlier before 16 weeks. So here's the result showing if you start the aspirin after 16 weeks, the efficacy is quite low. So it must be before 16 weeks, and also the dose is also an important factor. So the dose must be more than 100 milligrams, otherwise there's no effect at all to prevent preeclampsia. So this trial have been, was conducted in UK and in European. So in order whether to test the low-dose aspirin can also prevent preeclampsia in Asian country. So Professor Leonard also carry on this trial in different country in Asian country. So the result is the same. They can also prevent preeclampsia in different populations. Then the next question is how this aspirin work to prevent this hypertension during pregnancy. So then going back to the pathophysiology of the preeclampsia is mainly because of the trophoblast invasions is defect during the placenta development. And then afterward, there's many apoptosis happens in the placenta and induce a lot of toxic effect, toxic markers, biomarker, and also even releasing DNA from the placenta to maternal circulation trigger a lot of inflammation reactions. So and then the aspirin effect actually, how does it work? So then we have to go back to the pharmacology of the aspirin have been known for 100 years. So one of the mechanisms is COX-1 inhibition. So mainly try to dilate the vessels to improve the placenta circulation assumably. And also the second mechanism is COX-2 acetylations that relate to the inflammations. So aspirin can reduce the inflammations and then through this COX-2 acetylations. And also probably also improve the oxidative stress and the inflammation apoptosis in the placenta as well. So in order to show this mechanism of aspirin in our placenta, so people studying the clinical study to see whether is there any marker can be reduced by the aspirin during the pregnancy. So two of the markers have been well known to predict the preeclampsia is SLIT. SLIT is an anti-angiogenesis factors during the pregnancy, and PLGF is placenta growth factors related to the growth markers for the placenta. So after aspirin, there's a data to show they can improve the PLGF secretions and lower the anti-angiogenesis SLIT secretions as well. So And then our Asia trial also confirmed this result by showing that the PLGF is increased and SV is decreased. And then people also look at the uterine circulations, blood flow and resistance as well. So the early trial doesn't show any improvement in this vascular circulations. But for our Asian trials, we do show the aspirin can improve the circulation measured by ultrasound finding. So based on these three clinical trial, and then we showing that low-dose aspirin can improve the vascular circulation system by inducing vascular dilations and improvement of the circulations. But however, is there any other mechanism we are very interested? So based on this, in order to find the mechanism, we focus on the metabolism of the placenta. So we do the metabolomic of the precancer patients. And first of all, we confirm that the aspirin effect had been compliance rely on the report by the patients. We use the metabolite markers to confirm that the patient do take good compliance of the aspirin. And then we also look at the metabolic clock evidence during the pregnancy as well. So based on this study, they have identified some marker based on metabolomic marker can really predict the placenta maturations. So based on this marker, we going back to look at how this aspirin will effect on this metabolite. And our result showing that the aspirin can really slow down the placenta aging. So that means to try to avoid the preeclampsia develop so early. So this is totally matched with our original hypothesis for how this preeclampsia developed. So no matter the pregnancy extent, eventually the mother will develop preeclampsia. So the aspirin can move on this the biological clock to further delay the development of preeclampsia. So this is how we hypothesize the aspirin can improve the aging and metabolism of the placenta. So, and then the improvement probably just for 1 to 2 weeks, but however, this 1 to 2 weeks already significantly improved the clinical outcome of the particular for the baby in terms of the maternal death, neonatal death, and also SIGR, etc. So the next question, yes, we know that the aspirin can improve the metabolism in the placenta, but how this metabolism affecting the placenta growth and placenta functions? And then this raises up further questions of our study. So in order to study, we're going back to the pharmacology of the aspirin and also the basic of the pharmacology study, the AD and AMDE, etc. So, and then we're going back to see the aspirin metabolism pathway. There's many enzymes related to the aspirin metabolism, including BCHE, CYP2E1, and UGT1, and then also GLAT as well. So we focus on this enzyme because we believe that there's something wrong with this enzyme, probably affecting the efficacy of the aspirin. So my PhD students look at this metabolism in different ways. So the first way we look at the pharmacogenomic to see whether is there any mutation related to this failure of the aspirin treatment. The second thing is the PK and PD of this the enzyme in the circulation and also in the placenta. And lastly, to see how the gene expressions of this metabolism enzyme in the placenta as well. So first, the result of the pharmacogenomic. So we screen all about 250 aspirin-related enzyme, and then we cannot see any genome-wise significant mutations, variants related to the failure of the aspirin. So then we exclude this the effect But however, we also extend our study to the exon sequencing, and then we, based on the limited sample size, we identify some interesting sleep that relate to the circulation development and vasculature development as well. So we will further to extend our sample size to try to identify is there any important sleep as well. Then next thing is the pharmacokinetics. So pharmacokinetics, we look at the circulation, the aspirin enzyme as well, and then we follow on the metabolite of the aspirin, and then we did not see any differences of those patients response to the aspirin and those patients that didn't respond to the aspirin. And also in the placenta as well, they don't see any difference in this dose in the metabolite in different groups as well. And then the last thing, we have to go to the placenta metabolism. So we try to study the expression level of this different aspirin metabolism enzyme in the placenta. And then we identify two enzymes is actually differentially expressed. And then particular one of the enzymes, GLAT enzyme, actually is highly expressed in those patients didn't respond to the aspirin. And then we try to see whether is there any prediction value of this enzyme in the placenta can predict the preeclampsia outcome as well. And then you can see there are significant high risk if the gene expressions is different compared with the well-established clinical outcome as well. And then the next thing is if the enzyme actually is dysregulated, so how does it affect the placenta functions? And then we look at the placenta angiogenesis information, senescent marker, and different information marker as well. And then we find the significant correlations of this enzyme expression level with the different placenta functions as well. So this further confirmed that the different aspirin metabolism actually do affecting placenta functions and then further develop the preeclampsia at the end. And then we use the in vivo model to confirm. We use the placenta trophoblast cells and try to overexpress and downregulate the GLYT aspirin metabolism enzyme, and then study the expressions of the inflammation, angiogenesis, vasculatures, and different enzymes. Confirm the in vivo result as well. So basically, our finding confirmed that there's no major effect of the genetics and also PKPD related to the response to the aspirin. But however, the placenta expression level of the enzyme actually do affecting the effect of the outcome after the treatments. So here's the result actually published in Circulation Research in this year, last year. But the next thing is whether we can identify new therapy, try to prevent the preeclampsia further. So based on our metabolomic study and aspirin results, so now we are trying to combine both aspirin and also metabolism by the metformin and try to see whether there's a combined effect and synergistic effect to improve the clinical outcome. So we do the in vitro study and try to combine aspirin and metformin, and then here the results showing that the combined effect actually can further improve the trophoblast functions. And also in vivo, also the blood pressure is significantly lower, and also the complications of the mother and the fetuses and also reduced significantly. So right now we're doing the large scale clinical trial and trying to combine the aspirin and also metformin. And hopefully we will finish this trial in this year, and then we will have a result next year, try to report to you. So I would like to thank you for your attention, and this is our team. Thank you very much. Yes.
Thank you so much. That was a very insightful overview over a huge amount of work. I learned a lot. We're beyond time, but we should allow ourselves one or two questions. All crystal clear. Well, if that's not the case, then thank you again.
Thank you very much.
Thank you, speakers and moderators. Please come together for a group picture together. So may I invite Dr. Nuzinovic, Professor Tang, Professor Kong, Dr. Owen Ko, Dr. Zugimura, and Professor Wang to come together for a group picture. So thank you to all our brilliant presenters and distinguished moderators for bringing the session to such a perfect close. And we hope the inspiration sparked today will ignite new collaborations across our expert community and accelerate groundbreaking discovery. So please look at our photographer. All right, with big smile. So we wish for even more innovative technologies and insight to emerge, ultimately benefiting more patients and advancing cardiovascular science worldwide. Do we need a thumbs up? Yep. All right. Thank you so much, ladies and gentlemen. Thank you so much. Please take a seat. Now we have come to the exciting prize presentation session for our Young Investigator Awards and Best Poster Awards. Young presenters prepare their research poster or oral presentation with tremendous care, pouring their research passion onto paper and engaging in meaningful exchange with their fellow attendees. Before we announce the winners, may I now invite Professor Ralph Adams to give a few words on the performance of this year's posters presenters. Professor Adams, please. Just a few words.
Thank you very much. Yeah, it was a fantastic meeting, and the posters were just as amazing. The only thing I wish we would have more time to stand at the posters and discuss with the young colleagues who did this amazing work. But I was really very impressed by the quality of the data and the amount of results shown. So that's really great. And congratulations to all of you who presented or who prepared a poster for this event. Thank you.
Thank you, Professor Adams. Please take a seat. And we would also like to thank our judges, Professor Sean Wu and Professor Nicholas Smart, for co-judging the poster award. And apart from the poster presentation, 5 young investigators have presented their exceptional hard work on stage this morning. We believe everyone gained valuable insights from this interaction. Our judges have also had a challenging time assessing their presentations. May I now invite Professor Thomas Brown and Professor Wang Yibing to give a few words on their performance. A round of applause.
Yeah, I think all 5 actually deserved a prize, but we had to select 3 of them. But you did really amazing work. So really, all the judges we actually recruited 2 additional ones, were really impressed by your presentation. So congratulations. So we had to select 3. I think, Bin, you have the names. Actually,
yeah, I think they're going to announce the names by Zhou Bing, but I just want to echo Thomas' comments that the initial grades, scores for 5 speakers are exactly the same. I'm not kidding. It's never seen such a random distribution. That means that every one of you is a winner. You actually captured with your accomplishment, being recognized with your strengths and with your accomplishment by our reviewers. And really, I'm so impressed. And this is the part of the conference I always feel very motivated, but also a little bit depressed, because you are so good. And it almost posed a threat to us, actually. But we're very, very excited for you. We're happy for you. And hopefully this, whether it's winning or participating, is actually just a first step into the real science and for a very successful future. So thank you.
Thank you very much, Professor Brown and Professor Wang. Please be seated. And we would also like to take the chance to thank our judges, Professor Wang Dazhi and Professor Xu Qingbo, for co-judging the award. So what are we waiting for? Without further ado, to present the awards, please join me in welcoming Dr. Nuzinovic, Senior Editor at Science Magazine, to the stage to present the well-deserved prizes. All right, so for the Best Poster Awards, we have 3 outstanding winners. So the winners are announced in their order of surname. So the first outstanding poster presenter is, drumroll, Ms. Li Jie. Congratulations. Please come to the stage for our group picture with Dr. Nuzinovic. Yeah, let's come to the front for our group picture. Congratulations to everyone, because the score must be like really tight in this case. You just come together, hold the, yeah. Thank you, Li Jie. Please take a seat. And I will announce the second outstanding poster presenter. And her name is, drumroll, Ms. Lin Jia Li. Congratulations. Very precise. Perfect. Thank you so much. And I will announce the third outstanding poster presenter. And all right, drum roll. And this is Dr. Luo Minghao. Congratulations. So thank you so much. And this is the announcement for the outstanding poster presenter. So Doctor, please stay. Oh, please stay to continue. Yes, to present for the Young Investigator Award. So I have the result in my hand, and the award is announced also in their order of their surname. So the first outstanding Young Investigator Award goes to Mr. Hou Yangfeng. Congratulations. Brilliant. Thank you. So this is the first winner. And then we have our second winner. Our second winner is Miss Li Yangxian. Congratulations. Thank you. And then we go to the third winner, and she is Dr. Zhang Mingjun. Congratulations. I can see all the unbelievable faces on our winners. Thank you so much. Thank you so much and congratulations. Please take a seat. Please take a seat. Thank you so much. To bring our event to our gracious close, may I now invite our distinguished co-chair to deliver their concluding remark. Please welcome Professor Zhou Bing, co-chair of this symposium and professor at the Chinese Academy of Sciences, to speak for us. A round of applause.
Thank you. As we bring this 2 days cardiovascular conference to a close, I would like to extend my gratitude, appreciation to all the distinguished speakers and the participants who have joined us, come across from Asia and also from US, Germany, UK, and beyond. In the past 2 days, we have had the privilege of hearing the latest research from those leading experts in the world. I think the diversity of perspectives, as well as the depth of knowledge shared, have been truly remarkable. I think we have all enjoyed those stimulating discussions and new ideas throughout the talks, and outstanding contributions from young investigators in their talks and also in their poster presentations. Together, we have learned a lot of cardiovascular research, latest advances in the cardiovascular biology, and also in the cutting-edge technologies. I think this will encourage us to inspire new research directions, as well as build new cross-border collaborations after today's meeting. On behalf of the organizing committee members, I would like to thank the Croucher Foundation and the Young Academy of Science of Hong Kong, and also all the supporting teams, especially from Kathy's team members and others from YSHK, for their efforts and meticulous planning that made this conference possible. Finally, I'd like to thank each of you for your valuable contributions. I wish you safe travels and look forward to our paths crossing again in the near future. I wish you all happy and wonderful weekend. Thank you.
Thank you so much, Professor Hou. Please take a seat. Please take a seat. And finally, please join me in welcoming Professor Anderson Chum, President of the Hong Kong Young Academy of Sciences. A round of applause.
My name is Anderson Chum, so it's a real pleasure to be here with all of you. So distinguished guests, esteemed speakers, colleagues and students, as the president of the Hong Kong Young Academy of Sciences, it is my great pleasure to deliver this closing remarks alongside our Professor Zhou Bing as we conclude this extraordinary 2 days here at CU Hong Kong. When we set the theme for this symposium as the cardiovascular sciences, cutting-edge technologies, our goal was to highlight the innovative frontier of cardiovascular sciences. Over the past 2 days, the goal has been thoroughly exceeded. From tracing the origin of de novo coronary collateral formation to the latest advancements in cardio regeneration, the depth and caliber of the science presented have been truly phenomenal. I want to extend my deepest gratitude to our co-host, co-chairs, Professor Zhou Bing and Professor Kathy Lui, as well as the entire organizing committee for assembling such a thought-provoking program. We also owe a lot of debt and of thanks to our sponsors, ATCG Limited, Beijing Yiren Technology Company Limited, BioLink, Miltenyi Biotech, and Shanghai Model Organism Center. This conference wouldn't have been possible without the contributions of our supporting organizations. So our special thanks go to City University of Hong Kong, Hong Kong Baptist University, Hong Kong Metropolitan University, Institute of Professional Education and Knowledge, Lingnan University, St. Francis University, the Chinese University of Hong Kong, the Education University of Hong Kong, the Hang Seng University of Hong Kong, the Hong Kong Polytechnic University, the Hong Kong University of Science and Technology, the University of Hong Kong, Hong Kong Applied Science and Technology Research Institute, Hong Kong Cyberport Management Company Limited, Hong Kong Science and Technology Parks, Hong Kong Young Industrialists Council, the Hong Kong Academy for Gifted Education, World Association of Young Scientists, a long list of our supporting organizations. I would also like to thank all our speakers from across the globe. Your willingness to share your pioneering work and unpublished data has not only elevated our scientific discourse, but also reinforced the global spirit of collaboration that is essential for tackling the complex problems of cardiovascular diseases. In Young Academy of Sciences of Hong Kong, one of our greatest passions is nurturing early career scientists. That is why I was particularly inspired by this morning's Young Investigator Awards section and the poster presentations outside. Your innovative approaches, whether it's about decoding the mechanism of cardio regeneration initiated by immune cells, or utilizing nuclear features for cardiomyocyte identification, prove that the future of cardiovascular science is incredibly bright. Congratulations once again to all the prize winners. Thank you all for your active participation and your dedication to pushing the frontier and boundaries of science. I look forward to seeing you all again at our upcoming events and continuing this exciting academic journey together. Have a nice day, and thank you all once again.
Thank you, Professor Shum, for the encouragement. Please be seated. Ladies and gentlemen, this concludes the Cardiovascular Sciences Cutting Edge Technology Symposium. Thank you for 2 exceptional days of groundbreaking science, inspiring awards, and meaningful collaborations. Our heartfelt thanks to the Center for Excellence in Molecular Cell Science, Chinese Academy of Sciences, CUHK Joint Laboratory for Cardiovascular Sciences, Croucher Foundation, and the Hong Kong Young Academy of Sciences, Tencent Charity Foundation, and the Innovation and Technology Commission for their support, plus all the speakers, moderators, and participants. A round of applause to all you all. May today's insights spark future breakthroughs in heart health. For guests who are invited to luncheon, please follow the instruction from our helpers to the venue, and we look forward to seeing you at the next Croucher and YASS event. Thank you, and have a great weekend. See you all.